Glycolysis is a series of reactions that ultimately transforms glucose into energy molecules known as ATP that are used by the cell. Now, the net result of glycolysis is the formation of two pyruvate molecules, two ATP molecules, two NADH molecules, two water molecules, and two hydrogen ions. Now, glycolysis is an anaerobic process, and that means it does not require oxygen to actually take place. And that's why glycolysis is used by all different types of cells, including prokaryotic and eukaryotic cells. Now, glycolysis can be divided into three stages. We have stage 1, stage 2, and stage 3. Now, in stage 1, we actually use energy. So the entire purpose of stage 1 is to prepare the glucose molecule for breakdown. So in stage 1, we trap that glucose inside the cell and we transform our low energy glucose into a high energy glucose. And then in stage 2, we use that unstable high energy glucose and we break it down into two molecules, and then we use those two molecules that we broke down in stage 2 in stage 3, and we use those molecules, we harvest the energy within those molecules, and we produce the ATP molecules in stage 3. So let's begin by looking at the details of stage 1. So in stage 1, we take the glucose, we transport that glucose across the cell membrane into the cytoplasm of the cell, and then we use an enzyme known as hexokinase to basically transform a glucose into glucose 6-phosphate. So we use an ATP molecule, we take a phosphate group from the ATP molecule and transfer it onto our glucose. So we form glucose 6-phosphate. Now, there are two reasons why we have to phosphorylate our glucose, why we have to add that phosphate group. One of the reasons is because we want to trap that glucose inside the cytoplasm. We want to keep that glucose from exiting our cell. And by adding this negatively charged phosphate group, we basically keep that glucose inside the cytoplasm because it no longer is capable of being transported across our cell membrane. So once our glucose is transformed into glucose 6-phosphate, it stays within the cytoplasm of that cell. The second reason why we have to phosphorylate our glucose is to transform the glucose from the low energy stable form into a less stable form. So we destabilize the glucose by adding the phosphate group. So we create this negatively charged group on the glucose, and that destabilizes our molecule, and that will help us break down the molecule in stage 2. So step 2 of stage 1 involves transforming the 6-membered sugar glucose into the 5-membered isomer, the fructose. So we use an enzyme known as phosphoglucose isomerase, and we basically transform glucose 6-phosphate into fructose 6-phosphate. So step 2 does not require energy. And in step 3, we take the fructose 6-phosphate and we use an enzyme known as phosphofructokinase. We use an ATP molecule and transfer a second phosphate group onto that fructose. So fructose 1,6-biphosphate is formed that contains 1,2 high energy phosphate groups, and we also utilize that ATP molecule. So this is a very unstable molecule compared to our glucose, and that's exactly why in stage 2 we can now break down this unstable molecule, as we'll see in just a moment. Now, step 3 is actually very important in stage 1 because it's our committing step. So once we undergo step 3, our glucose molecule actually commits to our glycolysis pathway. So for example, if we move back to this molecule, the glucose 6-phosphate, the glucose 6-phosphate does not necessarily have to actually go on to produce the fructose 1,6-biphosphate. If we have plentiful amounts of energy inside the cell, the glucose 6-phosphate can then be stored as glycogen. Now, if we don't have that much energy in a cell, then the glucose 6-phosphate will go on to form the fructose 6-phosphate, which will go on to form the fructose 1,6-biphosphate. But once this step takes place, that glucose is fully committed to our glycolysis pathway. That is, there is no turning back. So once we form the high energy fructose 1,6-biphosphate, we move on to stage 2. In stage 2, the entire purpose of stage 2 is to basically take the high energy molecule and break it down into 2 3-carbon molecules. So by using an enzyme known as aldolase, so we basically transform the fructose 1,6-biphosphate into the dihydroxyacetone phosphate, this molecule, and the glycerol aldehyde 3-phosphate, also known as PGAL, which looks something like this. Now, in stage 3, we actually need to use the PGAL. So in stage 2, the second step, the 5th step, is to take the dihydroxyacetone phosphate and transform it into the PGAL by using an enzyme known as triose phosphate isomerase. So in stage 2, we basically do not use any energy, and the entire goal is to produce 2 PGAL molecules. And finally, let's move on to step 3, or stage 3. So stage 3 itself involves 5 individual steps. And this is the process where we produce our ATP molecules as well as the water molecules and our NADH molecules. So let's begin with stage 3, and we begin with the PGAL molecule that we form in stage 2. So we take the PGAL and we basically react it with a special type of enzyme known as glyceraldehyde 3-phosphate dehydrogenase. And this enzyme basically uses an NAD cofactor as well as a PI, which is our phosphate, and it produces the 1,3-biphosphoglyceride. So basically, we reduce the NAD plus into NADH by taking this H from the PGAL and transferring onto the NAD. And then we take that phosphate and we add it onto this carbon. So the entire purpose of step 6 is to basically reduce our NAD and oxidize this PGAL. And so basically we add a second phosphate group and we create a destabilized molecule. Now this 1,3-biphosphoglycerate is also known as 1,3-BPG. And then we take the 1,3-BPG and we actually mix it with ADP as well as an enzyme known as phosphoglycerate kinase, and we transfer this phosphate group onto the ADP to produce our first ATP molecule, as well as the 3-phosphoglycerate, which also contains this phosphate group. Now, in step 8, we take this and we transform it into this molecule 2-phosphoglycerate by using an enzyme known as phosphoglycerate mutase. And what we do is we transfer this phosphate onto this section as shown. And then in step 9, we have an elimination step taking place. So we add an H onto this oxygen, and then we basically kick off that water molecule that is produced as a result. So in step 9, we basically have a condensation reaction taking place. We produce the water and the phosphoenol pyruvate. And finally, in the last step, in step 10, we also produce an ATP molecule by transferring the phosphate onto the ADP. And the final step, the final product, is our pyruvate molecule. And the enzyme involved in step 10 is known as the pyruvate kinase. So the final product of stage 3 and glycolysis is pyruvate. Now notice in stage 3, in step 6, we produce 1 NADH. In step 7 and 10, we produce 1 ATP each. And in step 9, we produce a single H2O. Now this only shows 1 PGAL reacting, but in stage 2, we produce 2. So that means stage 3 takes place twice, and we form 2 NADHs, we form 4 ATPs, and we form 2 H2O molecules. So now let's count up all the reactants and all our products. So the reactants are basically the glucose. So we also have 2 ATP molecules, 1 and 2, that have to be utilized. We have 4 ADP molecules. So we have 1, 2, and because it takes place twice, we have 4 ADP molecules. We have 1 because it takes place twice. We have 2 PIs, phosphates. We have 2 NAD pluses because this takes place twice. And we have 2 H pluses, which I haven't shown in these steps. And the products are 2 pyruvates, once again, because this takes place twice. We have 4 ATPs, so basically 1, 2, 3, 4, because it occurs twice. 2 NADHs, we have 2 H2Os, we have 4 H pluses, once again I haven't specified where those were formed, and we have the 2 ADPs. So now if we cancel out some things, so basically we have 4 H's here, 2 H's here, that crosses out, this becomes a 2. We have 2 ATPs, we have 4 ATPs, this crosses out, this becomes a 2. And we have our we have the 4 ADPs here, the 2 ADPs here, these cross out, this becomes a 2. So the net reaction of glycolysis is as follows. We have a glucose, we have 2 ADPs, 2 phosphates, and 2 NAD pluses on the reactant side, and we produce the net result are 2 pyruvates, 2 ATPs, 2 NADHs, 2 water molecules, and 2 H plus ions. So basically, stage 1 of glycolysis requires 2 ATP molecules, and we produce our 2 ADPs. In stage 2, we take the high energy fructose 1,6 biphosphate and we break that down into essentially 2 PGAL, and no energy is actually used up or produced. And in stage 3, this is where we produce our ATP molecules. So we produce a total of 4 ATP molecules, 2 NADHs, 2 water molecules,